Thank you very much for the introduction. I would also like to express my gratitude to the organizers for giving me the opportunity to share my research. It's a great honor for me to be here today to present my PhD work on artificial intelligence method for investigating the internal organization of hepatoblastoma tumor tissue in the liver. So I'm a mathematician from Mathematical Institute of Bordeaux, currently starting my third year of my PhD under the supervision of Baudouin Denis de Sénéville and Christophe Grosset within the Monk and Mircad teams in Bordeaux. So in this presentation, I will first introduce our objectives and the microscopy data we use. Then to study the internal architecture of tissues, we need to segment and identify each individual biological elements such as nuclei, cells, or even nucleoli. So to this end, we employ deep neural network, and I will explain their general functioning and share some example of results. And next, I will focus on more complex scenario as cell segmentation in volume images, and I will provide an innovative approach dedicated to our specific challenge. And finally, using this segmentation, I will present some biological insights that we can develop by computing bioarchitectural parameters. So now, before anything else, why is it so interesting to study the tumor tissue architecture? So when building with Legos, the way we arrange the pieces leads to different construction. And similarly, in biological tissue, the 3D orientation and organization of various organelles results in different structures. And the study of these structures leads to a better understanding of the mechanism and function within the tumor. So to achieve this, we acquired at least 1,000 slices in Hepatoblastoma PDX model using SBF-SEM. And moreover, the acquisition parameters written here allow us to capture a substantial amount of information about various biological elements. And to study them, we need to perform an accurate segmentation and labeling of each individual biological element. So first, some studies analyze a few manually segmented cells and organelles in tissue sample, like in this video. However, this does not allow us to study a large volume in 3D, as the one on the left, because we need automatic segmentation methods. So for this, we leverage the recent advancements in deep neural networks. So now let's explore how supervised learning deep neural networks work. And one of the best analogy we can draw is how can we learn to ride a bicycle as a child. And no matter how hard I try to explain how to do it and how to balance, it's by trying and sometimes by falling that the child learns to ride bicycle. So it's a trial and error learning system, and that's exactly how supervised learning works. So for instance, if I want to segment all cell nuclei in electron microscopy image, I will input the raw image in the input of my network here, and I will compare the prediction with the ground truth. So this step is known as forward propagation, and the ground truth is the manual segmentation of all in the image by a biologist, sometimes assisted by a computer software. And to assess the accuracy of the prediction, we compute a loss function, which we aim to minimize. And then an optimizer recalculates neural network's weight to reduce the loss score. And this adjustment process is known as backward propagation. And by iteratively applying forward and backward propagation on different images, the neural network will gradually reduce the loss score, allowing the neural network to segment new images with increasing accuracy. So with this method, we can directly link the input, the raw image, electron microscopy image, and the output, the predicted segmentation. And this is what we call end-to-end strategies. So in the following, we employ the NNUNet neural network. It is a publicly available and state-of-the-art framework for various biomedical image segmentation tasks. And however, unfortunately for those without programming skills, there is no graphical interface to use the NN unit with click button interface. But using this, we can achieve excellent results as segmenting nuclei, nucleoli, lipid droplets, or even blood capillary, represented here in red. So in the first row is a 2D visualization of segmentation, and the second row is a 3D visualization obtained after segmenting all images in stack. However, cell segmentation in volume images is a specific challenge that has been addressed in the literature and requires dedicated strategies. So currently, there are automatic methods such as cell pause and semi-automatic methods such as microscopy, which is derived from the segment anything model of the Meta company. And these approaches are well known in the biology community since they were implemented in a Napari plugin for user-friendly interface. So now I'm going to present you an innovative and automatic cell instance segmentation approach that significantly outperform state-of-the-art methods in our context. So as we can see on this video, in our context, cells are large and tightly packed, and moreover, they contain various biological elements with different grayscale texture. So it is more, for this reason, it is more efficient to initially segment cell contours than cells directly. We can train a deep neural network to segment cell contours. Here again, we employ the 3D architecture of the NN unit. And after the segmentation, we apply a connected component algorithm to identify each individual cells to compute some parameters and compare them. But this approach yields poor results that require tedious and time-consuming manual correction for the biologist. So as we can see here, the colored cells are those that are correctly identified compared to the ground truth, and all black cells contain errors. We have represented in the red area the blood capillary and an hemorrhagic zone. And we can observe that most of the correction involve detecting and filling gaps in a cell contour, as we can see here, for example, here again, or even here. So now I'm going to show you that we can improve this segmentation result by using an additional module, also based on deep neural network. So here we were inspired by what the biologists do in front of his tablet to correct the segmentations. So I mean detecting and filling gaps in areas with missing cell contours. So we called our dedicated network as cell contour closing operator, or COPNET, in reference to the morphological closing operations. And at this point, it is important to know that a network can effectively output a binary segmentation, as I explained earlier, but also a probability map where each pixel value represents the probability of being a cell contour in our context. So this probability map contains more information than the binary segmentation, and that's why we employ them here in the input of our COPNET. Now, to address the limitation of training data availability, we generated a synthetic cell contour probability map with missing information areas with two approaches. The first one is based on a partial differential equation with the ground truth segmentation as initial guess and dedicated boundary conditions. And the other one is based on semantic image synthesis by a generative adversarial network, a GAN, developed by NVIDIA. And the randomly located red areas in the input here indicate where signal drop are generated by the GAN to create missing information in the probability map. So the corresponding probability map are shown here. And in the following, I'm going to present the results obtained by using the first simulation method, but similar performance can be obtained by using the GAN-based simulations. So we can see that by using our closing operator, the number of correctly identified cells has significantly increased on this slice. And moreover, when tested across a set of 20 images from a different tissue, the proportion of correctly labeled cells has increased from 24% to over 70%. And we have also compared our approach with a state-of-the-art cell instance segmentation approach named CellPose, which only achieved 29% of correctly labeled cells. And finally, the centerline dice demonstrates that our approach reduces significantly the need for manual correction for the biologist. So at this point, I have demonstrated that there are end-to-end strategies for the automatic segmentation of various biological elements. And for more complex scenarios, we have seen the possibility of adding deep neural network module for improving the segmentation of specific elements, as we have seen for cell segmentation. Now we can use this segmentation to enhance our biological and medical knowledge. And as we can see on this video, a key biological insight will be to study organelle volume relative to their distance to the blood capillary. So here, the larger the organelle, the more yellow it appears, and the smaller it is, the bluer it becomes. So here's for nuclei, and just after for mitochondrial network. So now when examining the correlation between a volume of different structure type relative to the distance to the blood capillary, we can observe a negative correlation for cell and mitochondrial network, while no correlation for nucleus volume. Next, we can also compare the distribution of nuclei spatial orientation across 3 tissue samples from different patients. And we can observe a significantly different spatial orientation in the third stack. And we hypothesize that is because this stack contains a large hemorrhagic zone, which reduces pressure on the tissue and leads to a different spatial orientation of nuclei in this tissue. So in conclusion, we have seen an automatic method for segmenting biological elements. Some of them are generalists, such as CellPose and NNUNet, and others are specialists to an organelle, such as our proposed cell contour closing operator for cell segmentation. There also are semi-automatic methods, such as microsam, and the combination of these two approaches, automatic and semi-automatic solutions, aims to reduce the need for the biologist to interact with his tablet to correct the segmentations. And this makes it possible to carry out bioarchitectural analysis of tissues, such as determining new features, organelle organization in 3D, comparing different tumor tissue, identifying different cell types, or making links with clinical data. So this enhances our biological knowledge and can lead to improved diagnostic and treatment planning, ultimately enhancing our patient care in oncology. So thank you all for your attention. I would also like to thank all my collaborators for their contribution to this multidisciplinary project at the intersection of applied mathematics, cancer biology, and electron microscopy imaging. And so now I'm happy to answer any of your questions.
Thank you very much. Are there any questions? I had a question for you. So in connectomics, it's quite popular to use affinity-based voxel predictions as a basis then for watershed-based segmentations. Is that something you've also looked into?
Yeah, I use a watershed for the connected component algorithm to identify each individual cells based on contour segmentations. To take into account that even if there are 1 pixel of error, and if 2 cells are connected with 1 or few pixels, we can separate them with a different label. It's more flexible than just look if they are connected.
Nice talk, thank you. You mentioned that you're using this NN UNet, right?
Yeah.
And what is basically the difference between with the normal UNet, or what's the benefit of that particular architecture?
Yeah, it's a framework which adapts many parameters of a classical UNet and training UNet parameters. So it adapts the architecture of the UNet, for example, patch size, etc., to our data, to the size of our image, to our computer, and it's by using this framework, we have no tuning to do. We just give the data, and it adapts all parameters to obtain the best segmentation.
So it's basically a framework to adapt UNet architecture to particularly to your type of data, right?
Yeah. We don't have to create 2D architecture. For example, if you want to use 2D or 3D, it's already included. And this NN UNet achieved best wins many international competition of segmentation.